Quinn, you listen to the first episode and you realize that we say, welcome to Orders of Magnitude, the podcast in which we talk about science from the smallest X to the biggest Y. And most of the time, it's contextual to what our guest is going to talk about. For example, with proteins, we said from the smallest amino acid to the biggest macromolecule. Do you have any idea of how to describe what we talked about today from the smallest X to the biggest Y? Putting you on the spot.
Probably I guess from the smallest virus to the biggest epidemic.
Oh. To the biggest epidemic?
Nice, that's nice.
Okay, I thought about this because I had a whole day.
Is that the one in your head?
From the biggest I would have said like from the tiniest genome. Because viruses are the tiniest genomes, and I like it when people don't understand at the beginning, and then they listen to the podcast, and then they're like, I'm smart now.
So the tiniest genome to the largest
epidemic.
Oh, okay.
Do you want to do it?
Yes. Well, hello everyone. Welcome to
not too loud.
Sorry.
Let's start over.
Hello everyone, and welcome to Orders of Magnitude, the science podcast, where we explore topics going from the tiniest genome to the largest epidemic. Today we have Quinn with us, a PhD student studying biochemistry.
Correct.
At McGill University, and he studies virus, more specifically the Zika virus. And today we will talk about the Zika virus and viruses in general. What else did we talk about?
We did another episode of mental health. This is up and coming in 2 weeks, probably, or a bit more than that. But however, we talked about how the Zika virus structure can affect its function. It was very interesting. We'll get you guys to the point at which you can understand what a virus structure even is. And let's just add that also Quinn is my BGSS president. Yes. So I was nice to him all throughout the day because of power.
Power? How much power do you have?
Enough.
Nice.
All right. Do you have anything to add?
That's good. You're getting some brownie points, Phil.
Perfect. Have a good podcast.
That works. Well, Quinn, welcome to the podcast.
Thanks for having me, guys.
Thank you very much for coming. So today we're talking about viruses, a little bit of your research as well. Let's start by the basics. Why don't we start by talking what are viruses?
What are viruses? Yeah. So, I mean, the textbook definition, I'll give you first. Nice, we can break that down a little bit. You consider them an obligate intracellular parasite.
Ooh.
And that's three very complicated words.
Yes, exactly. So let's take that one at a time. Firstly, obligate. In order for the virus to replicate, it needs its host. Mm-hmm. When it's outside the host, it's just chilling, it's inert, can last a long time until it finds a host, but it needs that host to actually undergo its life cycle. Intracellular, the second part, the virus has to enter into the host cell, whether that's us, plant, bacteria, whatever. It needs to get inside so it can actually undergo its replication inside of another entity. And then finally, as a parasite, it uses pieces of that host in order to undergo its life cycle. Again, it can't do it on its own, and so most of the time it's not really giving anything to the cell, but it's taking the cellular machinery process is taking advantage of that in order to replicate and undergo its life cycle.
Very cool. Okay, so an obligate, which means that it needs some host to replicate, and it leads to all sorts of debates about whether viruses are alive. Intracellular, which means it needs another cell, and parasite, which means it basically parasites the cell in which it is.
Exactly.
All right.
Yeah.
That's already something. So we always see these little viral particle in the news. When we see COVID, we see this fuzzy ball with the spike protein sticking out. It's always the same color scheme for some reason. The ball is gray and the spike proteins are red. So you're saying that for most of its active life, a virus doesn't look like that.
For its active life, yeah, not really at all. That's how the virus would look after it's exited your cell and until it's entered again. But basically, when you look at that ball is basically just like a a shell that's guarding the genetic information. And in some viruses, they carry along some proteins or other information with them as well. But yeah, in the case of SARS-CoV-2, it just has the genome in there, and that's just a protective shell, and also has some utility for actually entering into the cell. But once it gets in, it just releases the genome, and then within the cell, I guess at that point, yeah, it does not resemble that virus whatsoever.
What does it look like?
It's whether RNA or DNA, just a string of genetic information, can vary quite a bit depending on the virus. And it's actively within your cell, either being translated using the host machinery to make its own proteins to undergo the various functions of its life cycle, or it can be undergoing replication. That's one of the steps that you need to go through. You basically enter in with your single copy of your genome, again, depending on the virus. And then from there, you need to make a number of other copies so that you can make that viral particle basically over again, 100 times fold, and release it so you can infect neighboring cells. And then just repeat that cycle.
Mm-hmm. Okay, so there are different types of viruses, right? Not all viruses look like a tiny ball with spiky things all around. Can you tell us about some different types of viruses?
Certainly. One of the other shapes of viruses, the ones I find kind of interesting, are the rhabdoviruses, and they make like a bullet shape. And that was one of the viruses I was working with in my master's previously. You also have...
They make a bullet shape, and then they shoot out of the cells when the cell explodes, and they go towards the other cell, or...
Not quite, but I really like the analogy. That's how I wish it came out, but not quite as dramatic as that. I guess viruses in general, like we think in these big villains, but they're just doing their thing, living, moving on to the next, and most of the time trying to do so as subtly as possible. And if that's what they're doing, then they're doing their job. Most viruses that we run into on a day-to-day basis, like, don't have any impact on our lives.
So the viruses that actually kill us are more of an exception. The virus has all of the advantage to just go through us, replicate, leave, and be super sneaky about it.
Exactly. And that's why most of the time, you'll actually see over time a virus will evolve to become less lethal. Because if it kills you, you can't spread it to your friends. Right? And so, yeah, like the virus that's able to transmit is much more dangerous than the one that's lethal. And I guess a good comparison is if you look at COVID versus Ebola.
Yeah.
Right? If you gave me a choice right now to be infected by one of those, it's not a hard choice. COVID any day of the week. I've got maybe like a 3% chance of surviving that, double vaxxed, higher now. Ebola...
3% chance of not of dying, you mean?
Yes.
97% chance of surviving.
Exactly.
Yeah.
Ebola is like the flip side. I believe the mortality rate at the height of that epidemic was 94%. Not fun, but it doesn't transmit very well. That was more so it transmitted in Africa due to cultural practices in terms of how they prepared and honored their dead. But when it spread over here to North America, like there were very, very few cases where there was actually transmission within North America. But if you got it, terrifying, absolutely terrifying.
So it's always that small optimization problem for the virus between replicating as much as it can without killing the victims too much, basically.
Exactly. Because, yeah, for the virus, I mean, it doesn't do any good to kill us. I mean, in most cases, it's the exact opposite. Like, it's kind of just an unnecessary side effect. Again, the virus itself is just a mindless thing. It's surviving, doing its thing, parasitizing from us. But, the side effects to us, it's already long gone. It's done its thing. It's not a pro or a con for it. It just, it is.
100%.
It's just doing its thing. It doesn't think, right?
Exactly.
It's just a bunch of molecules.
Yeah.
So, as you were saying, if it enters the cell, it doesn't look like a tiny evil guy inside of the cell. It's just DNA, like, flowing around. There's some proteins and things. Yeah. Right? Okay. So, we can't say, for example, that COVID is an evil virus killing people. It's just a thing in the air that is unfortunate for us.
Exactly. And unfortunately evolved to become super transmissible to us. And, that's just kind of the luck of the draw. And viruses, kind of by their nature, do evolve very quickly because they're just small pieces of genetic information.
Yeah, we have 3 billion letters in our genome. Once every generation, I think we have, 10 mutations or something like that. And this is because we have also in all the instructions to repair our genome. But the viruses, they have this tiny, tiny genome. So each mutation is a bigger burden or has a bigger effect on the virus, I would suppose. And It doesn't have the machinery to repair its own genome. So it must mutate very, very fast.
Exactly. And most of the time when you are infected by a virus, and this is a lot in the lab, like it's actually a population of viruses. Like you're being, I guess there's a difference between being infected and actually getting a disease. If we each ingested one SARS-CoV-2 particle right now, we would be infected with the virus, but we would clear that. You need a certain load and actually to see disease. But when you're exposed to, your hundreds, thousands, perhaps millions of viral particles, most of those are going to be subtly different, even if that's just at a single nucleotide level. Sometimes larger than that, you might have a deletion of a region and whatnot. And as long as it's not completely lethal to the virus, completely preventing it from replicating, it will persist at a low level. And sometimes there's some other, selective pressure that actually makes that different virus the one that is more useful.
Very cool. Let's back up a bit just to get all the details right before we pursue further. So, like we said, the human genome has 3 billion letters of DNA, basically. How small can a virus genome be? Let's say the one you're studying.
The one I'm studying is basically 11,000.
11,000. So that's like 30,000-fold less.
Many orders of magnitude. Yes.
Yeah, many orders of magnitude less. It's much less complex, so much smaller. And if you would have to do a numbered list or a bullet list of the steps that a virus has to take between the moment it is the small particle we see on the news to the moment it has successfully replicated in the host, could you make that list for us?
Yeah, I can walk you through that. So I guess I can use Zika as an example. It's the virus I study.
Yeah, you can. Yeah, let's do it for Zika.
And so, first off with Zika, you're either going to become infected by being bitten by a mosquito that's infected with the virus, or through having sexual relations with a partner that's infected. And once the virus enters you, it's looking to enter into your cells. And again, the virus itself, it's the genetic information covered by these structural proteins that are protecting it. In the case of Zika, it's a little bit less specific than some other viruses, and then we can kind of touch on that later. But those structural proteins basically interact with surface proteins that are on your cells. And in so doing, it's kind of like a lock and key, where all of a sudden the receptor recognizes the proteins on the virus, and is like, oh, that's something I should bring into the cell. And now that you've unlocked that, you internalize the virus, and then the virus actually breaks apart and releases the genome into the cell. In the case of Zika, the first step when it's in the cell is that it needs to be translated. And what that means is that it uses the host machinery to produce all of the viral proteins, which some of those are those structural proteins, again, to make new viral particles. And some of them are non-structural for the subsequent steps in the life cycle.
And again, for comparison, the human genome encodes for 20,000 proteins. How many proteins does the Zika virus encode for?
Zika has 11.
11. So that's very few information. So the virus has to be very smart about using that information to tell the host what to do.
Exactly.
It's almost just 1,000 base pairs per protein.
Yeah. Exactly. And most of those proteins actually end up serving multiple functions in the life cycle. You have to be hyper efficient.
Yeah, it has to be 11 Swiss knives, basically.
Exactly.
Okay, so far we have the small viral particles, which are basically a protective shield of keys. The key touches the cell, allows it to enter, and then it releases the genomic information, starts making the protein that is in the genomic information. What happens next?
So at this point, you've created your structural proteins and your non-structural proteins, and then basically they form a little compartment in your cells, and kind of take advantage of one of the organelles that's already there, makes its own compartment, and then the genome needs to replicate. And for Zika, that's a two-step process. So you start off with, it's considered a positive sense single-stranded RNA genome.
We'll get to that soon.
Yes. And for the replication, the first thing it needs to do is it uses those non-structural proteins to first make a complementary version of that strand, which is considered the negative strand intermediate.
It's like a carbon copy.
Exactly. And so from that negative strand, you can then make copies upon copies of that original positive genomic strand, which leave this little compartment and then are packaged up with those structural proteins you made earlier to make these new viral particles. And then those are transported out of the cell and are just released where they can infect the neighboring cells.
So it's almost like, what's the story, that Greek story, the Troy horse?
The Trojan Horse.
Trojan Horse, yeah. He comes and knock, knock, let me in, I'm friendly. And the cell takes it in.
Yep.
Because, oh, sure, it should come in. And surprise, surprise, just make more of myself for me. And so I can go, like, take your neighbors.
Guess what? It's my city now.
Exactly. Yeah. But the part is, it's like there's a lot less burning and pillaging.
Yeah.
It's a much more efficient process. You're kind of, the cell itself is less efficient because you're diverting some resources towards the virus. It's diverting towards itself. But, once it leaves the cell, the cell is still there. Mm-hmm. It's still functional, working. Other viruses, not so much. They can be more destructive on the way out.
Mm-hmm. Mm-hmm.
But yeah, in the case of Zika, that initial cell that was infected, it still is. It's not violent by any means.
That's interesting that you say this, that the cell is still intact, the cell is still there working. What happens with other viruses that make you sick? Why do you get sick if the cells are still fine?
So a lot of the times the symptoms you see are basically you responding to the virus. Mm-hmm. When you think about a fever, you're raising your body temperature. And that is, you're trying to basically increase the activity of your immune system against the virus. But if your fever gets too high, you have a lot of other issues, particularly in your brain and whatnot. Coughing, sneezing, again, you're trying to just, if it's a respiratory virus, remove those viral particles out of there. Mm-hmm. So a lot of the symptoms that you see are a result of that. It's basically just your body getting caught in the crossfire of your immune system trying to take out the virus. And again, the virus doesn't really care. As long as it can get in, get out, that damage that occurs is not a big deal. And
that's how its ancestors were selected.
Mm-hmm.
Exactly.
I'm sorry if I'm getting off topic. It's still on Zika.
Yep.
Zika, for example, I know that it has some developmental effects on babies if a pregnant woman gets Zika, for example, right? How does that happen if the cells are still intact?
So it depends. I mean, part of that too is that Zika can infect cells across your body.
Yeah.
And sometimes it can infect your neurons.
Mm-hmm. And
your nervous system is what's considered a protected part of your immune system.
Mm-hmm.
You don't want strong immune responses with your neurons because those cells, they don't regenerate. They're highly specialized. And so partially what can happen in those cells is the virus, as part of its life cycle, will induce some inflammation in that region. And again, that breeds an immune response, which can have negative effects on the nervous system.
Mm-hmm.
It seems as though it's a common theme amongst viruses to just sneak and seek shelter into the brain. I think COVID does it, herpes does it. I don't know other examples, but that's already like wide viruses from all different types, right?
Yep, certainly. And that's the thing, like that's the best viruses basically are just finding little ways to take advantage of your immune system. And finding a way to just kind of either evade it or to manipulate it to its own end. And it's kind of, again, this idea of a selective pressure, right? It's like once it gets into your body, that's what it's fighting against to stay alive. And so over time, if you have a random mutation that gives you a way to get a leg up on the immune system,
mm-hmm.
that's the virus that's going to win. You're selecting for that. And then similarly, in your immune system, the cells that can then combat that virus are also being selected for. And so it's an arms race. And neither side is ever fully winning. You really can't get to a point where you have a winner. And that's why these viruses keep evolving. And
it's like when two humans get into an argument and you can always reconcile opposing views, but nobody's going to get everything.
Yes, exactly.
Can we talk about how the rate of infection increases the rate of mutation? of a virus. How come if the virus can spread faster, there's a greater chance that we have a new variant of given virus?
Yeah, precisely. And so that's something that we saw a bit in the Zika epidemic, and particularly now with COVID and the variants. And basically, it's just a numbers game. So as we're kind of alluding to before, every time that genome replicates, there's a chance a mistake gets made. It's not perfect. And in the case of the virus, it doesn't have its own repair systems. And so it's more prone to have mutations arise. And so a virus that replicates to a higher degree in your cells, there's more chances for mutation to evolve. And every time it goes to a new person, there's a whole other now basically lottery that you go through.
And the generations are basically happening within hours rather than within 25 years for humans.
Exactly. And so it's just a numbers game. The more chances you get, the more mutations you're going to get. Some of those are going to be detrimental and it'll just die off. But you will have some that come in that allow them to outcompete. And, if you have a mutation, for example, and then the spike protein of COVID that allows it to get around the vaccine, that right now is a pretty big advantage with where we're at, especially in first world countries.
Yep.
So let me try to just make it a little bit more clear for listeners that are not used with terms like mutation or transcription, this type of things. It has nothing to do with the Hulk becoming green, right?
Yeah.
It's more like the genome of the virus. It's just a sequence of letters, right? Molecules that we have letters specific for them. And inside of human cells, or the cells of whoever is infected, there's this protein that is like a scribe, right? And it's just copying whatever the virus gave it to the cell. The virus gave to the cell this sequence and told the cell, copy this. The cell starts scribing and copying that text. The more text you copy, the more likely you are to make a mistake in your copy. And each one of those mistakes is a new mutation, right? Okay.
Exactly.
And then a mutation is what changes how the virus works.
Exactly. That's a great analogy for it.
Okay. Thank you.
Okay. So I think we've nailed down most of the general virus stuff. One small distinction I want to make, because it's a term that I don't even understand from what you said. Wow, it was a bit earlier, but we were talking about the life cycle of the virus. And you said that once the virus got into cells, there were some structural proteins and there were some proteins that were meant to replicate it. By structural proteins, you just mean basically the protective shell.
Exactly.
Okay, so early on, the virus just makes its own compartment somehow, using some of the proteins, I suppose. It replicates itself by making this negative strand, you said, or the carbon copy, and by using this carbon copy as a template to make more of itself. And this is also with non-structural proteins, you would say.
Yeah.
Is it with proteins from the host? Is the Scribe Matthias was talking about a minute ago, protein from the host?
So that's a viral protein, one of the non-structural proteins. But there are also host proteins involved all along. And each step, typically, at least when you're talking about Zika, they're playing more of a support role. But yeah, the main actors are these non-structural proteins, and that's the main difference. Is just basically, do you end up as part of the protective shell or not?
Okay, can we talk a little bit more in detail of your research? What do you do with Zika? What are you trying to study? On that?
So for me, actually, it's that negative strand carbon copy. That's the main thing that I'm interested in, and within biology in general, there's a huge focus on the protein level. Basically, you have your, at least in humans, DNA, which is your storage of genetic information. You make RNA, which are just copies of that are basically your recipes to make your proteins that actually do all the functions in your cell. And so a lot of time is spent on proteins and the functions that they do.
Yeah.
But Zika's genome is just a single RNA molecule. It gets into the cell. It has to be its own template for all of these steps. And what we know is actually that there are structures within the RNA. It's a single strand, and it can bind. Certain letters are able to bind to each other, and you end up with structural regions that actually have function. And we know for replication that there are structures that are necessary for recruiting the scribe in order to begin making your negative strand. but no one's really looked, at least for Zika, at the negative strand to see if there are similar structures to recruit that scribe to then make the positive copies.
Very good. When you talk about structure, I'm not sure if it's clear to everyone. So I think the structure people are most familiar with is the structure of DNA. We call the double helix a structure. And basically it's two strands that are just woven around each other. And this, in biology, we call this a DNA structure. In the case of RNA structure, you're saying, like you said really well, it's a single strand of RNA, the Zika virus genome. And some of the parts are woven onto themselves.
Yes.
And this is also a double helix, basically.
Kind of. So yeah, you basically create stems that are reminiscent of the double helix and also then loop structures off of that. And they do have a natural curvature to them, not exactly the same as the double helix of DNA. And part of it too is, you can have a structure that has a stem and it curves a bit, and then a loop, another stem off of that curves a bit. And so you can end up with these really weird structures. And sometimes they can even form a knot where the end of one loop has actually come back down to the single stranded part of the strand.
These very intricate structure, like you say, that are sometimes single stranded, so a random coil, sometimes a sort of double strand that we call a stem, sometimes stem loops or a stem followed by a loop followed by another stem, and it becomes super crazy, right?
Oh, exactly. Yeah, there's some really weird looking structures that we have no idea what they do.
So that's really cool, because what you're saying is the virus, Zika virus, comes, and we know that the genetic material of the virus, it uses itself to recruit stuff to make more of itself.
Exactly.
Oh, that's really nice. So it's not only proteins that can do this type of things. Basically, that's what you're saying.
Exactly. And that's something that I think probably just in the last decade or two, RNA in general has been seen as something that is multifunctional, even outside of viruses, but in particular in viruses, because again, we're dealing with just the single strand of RNA that has to do all of these functions. It's super cool how that's evolved.
And so you're trying to look at the carbon copy of the genetic material of Zika, and if this carbon copy actually does the same thing, if it can recruit the proteins.
Exactly.
Right? How would you do that?
Yeah, so first off, basically, we think that the way the Scribe works is that it starts at one end of the genome and then rides along, and the orientations that we basically call that is the 5' end is your left end and your 3' end is your right end. And to make your new strand, basically the Scribe comes at the 3' end and it makes that carbon copy that goes all the way to the 5' end. And so our thought, and where the research led a little bit thus far, is that at that 3' end of the negative strand where we think the Scribe needs to start, we think that there are structures there that we've identified that could recruit the Scribe. And the one that we're most interested in actually resembles the structure that does the same function on the positive strand. And what we're trying to do now is basically disrupt that structure in such a way that we don't think the Scribe would bind, and then see if, the virus is still able to undergo its life cycle, or even if we can, just take that single step outside of cells and see if we can, disrupt either through the structure or just the actual sequence, the letters themselves, see if those are important to actually recruit the Scribe.
It's so interesting. So in our genome, it's proteins that bring the Scribe somewhere, right? But the virus doesn't have the luxury of encoding for such a protein. So this RNA structure is a way to condense the information of the RNA. The RNA encodes to make protein, but it also encodes for like localization and interaction. And this is what basically you're saying right now.
Exactly.
And you're studying the way that at the ends it can recruit the Scribe protein, or let's call it by its name, RNA polymerase, RNA dependent RNA polymerase. Yes. It's something that reads RNA and makes RNA.
Exactly.
That reads the negative strand and makes the positive strand of RNA.
Precisely.
Okay. So you're saying that the way you study this is by disrupting one at a time the small letters of DNA. And for example, if you have a stem, you can disrupt this stem a little bit at a specific spot on the stem. And then you can see what happens to the virus replication?
Yep. So yeah, I mean, we can measure basically whether or not the polymerase is actually able to bind to the structure still, if we can disrupt that. And we can also look at, the output of our new positive strands being made, period. And then, kind of the holy grail when you're doing these experiments is that you want to disrupt this structure, and then you want to basically try to reform it, but using different letters, basically. And what that should tell you is, first off, if you disrupt it and you lose the function, okay, this whatever stem sequence, whatever is important. But if you're able to restore it, then you prove that it's necessary.
Yep. That's can we do a bit of a tangent on necessary and sufficient in science? You got a correlation. When you remove something such as the sequence or the stem, you lose the replication. But this could just be a correlation if you just observe DNA sequences. However, if you go in the system and intervene yourself and you're the one changing the stem and you lose the replication, now you can say it's something that is necessary. Is that a bit like it?
Pretty well.
And if you add a stem by yourself, you go in the system, you intervene, and all of a sudden you gain the interaction, you gain the scribe, the RNA polymerase being there, you say that this stem would be sufficient to recruit it.
Exactly.
And correlation plus necessity plus sufficiency means as close as we can get to causation in our ape system.
Yeah, exactly. Yeah, basically narrowing it down into the 99th percentile confidence that we actually think that this is occurring.
And how would understanding the structures at the end of the RNA molecule, how would understanding that be beneficial for development of treatments of this virus?
Yes, that's an important question. Zika in particular, there are no commercially available vaccines or antivirals currently, and so luckily there's not a high lethality rate with it, but it's something we really can't treat. And one option there, and one that people are starting to get into, is the idea of RNA structure-based inhibitors. So basically taking a really small molecule, and I've identified this structure that I think is important for recruiting the scribe. If we treat you with this small molecule that can enter your cells and binds to that specific structure, and this might be, just a small portion of it that's able to interact with. That might be enough now that the scribe doesn't have the correct orientation, the polymerase, to actually bind to it anymore. Mm-hmm. And if it can't do that, it's not going to replicate, not going to be able to spread.
And the reason why you're using a small molecule to disrupt that interaction rather than changing it is because you can't just find all the Zika viruses out there and mutate the stem, right? You can just go find all the... So instead, what you're going to do is once someone is infected, you find this little molecule that is super specific to the stem, and that goes and pluck the stem at the spot that the RNA polymerase scribe would have bound.
Exactly. Mm-hmm. And you hope that the small molecule that actually binds to that region is one that perhaps for, cancer or something else has already been starting to go through the validation approval process. Because that's really the big holdup. I'm sure, there are people out there who have libraries of these small molecules. I'm sure we could find one that could disrupt the structure. Fantastic, you're off to the races. But to actually get that to a point where you can get to human treatment is lengthy and expensive.
Yes, that was my next question, actually. If you have a molecule like this that can disrupt the replication of the virus, is there the possibility or the risk that this molecule will disrupt systems inside of the cell that we actually care for?
Certainly. And yeah, that's one of the important things to test. I mean, these small molecules, it's like they could also interact with proteins in the cell. They could interact with cellular RNAs. And so you can very easily have off-target effects, especially with something that's small. The specificity of it, can inherently be less, if you're trying to get something that's, a small pebble versus a boulder, it's going to be much easier to slot that in and interact with things. And
for the people at home, though, fear not, because this stem must be a very specific thing. It's not everywhere in biology that we have a stem that recruits the scribe like this, right?
Exactly.
And we have a bunch of chemists. There are chemists out there who work at making these specific molecules, but we're not quite there yet. But it's getting better and better.
Yes.
We used to be there trying basically randomly a bunch of molecules, and now, especially when it comes to targeting RNA, I think we're getting closer and closer to being able to target specifically some sequences.
Certainly. There's a couple groups that we've collaborated with that are really doing that on large scale libraries of these small molecules and characterizing the structures and specific sequences that they interact with. But yeah, then the question with these, and this is why you have to then go through your FDA approval process, which takes years, a lot of money, is like you have to validate that molecule isn't binding to some other cellular protein RNA.
Yeah, and you could validate all of that, put it in an animal, and you put it into humans, and it doesn't work all of a sudden, right?
Exactly.
For reasons that are out of what we can fathom.
Exactly. So if you're really lucky and can find, basically, you happen to find a small molecule that already has another use and has gone through this process, then you're laughing.
Repurposing a drug, you're happy.
Exactly.
Mm-hmm. Quinn, thank you very much for coming and telling us about viruses. It was a very interesting topic. Let us know how people can get in touch with you if they want to know more about your research.
Yeah. So, basically right now, doing my PhD at McGill in the lab of Selena Sagan. And if you look us up online, we have our website, and we'll probably post a link to this podcast as well.
Mm-hmm.
More personally, my email is just quinnabram@gmail.com. If you have any questions, I'm more than happy to answer them. I think you guys are doing a great thing here with trying to humanize science, and really get that out to the general public and communicate that, because there's a big disconnect there between the things we're doing day to day and the people in our lives who aren't scientifically literate.
Mm-hmm.
And I think that the more we can bridge that gap, the better.
Mm-hmm.
Beautiful. Thank you so much for coming.
Thank you for listening to Orders of Magnitude. If you enjoyed this episode, please leave us a 5-star rating on Apple Podcast and share the episode with your friends and family. If you would like to give us feedback, you can reach us at ordersofmagnitudepod@gmail.com. We would love to hear your opinions and ideas on the subject that we discussed today. Orders of Magnitude is an original project led by Philippe Carle and Matthias Schultz. The original music was composed by Vincent Ellis. Stay tuned for the next episode coming out in 2 weeks.